Hello everyone, I'm Nishtha Bansal, a third year MBBS student at APOL. I gave my NEET in 2020 and I secured a rank of 484 with a marks of 683. So today we'll be discussing about the chapter Microbes in Human Welfare, a chapter of class 12 biology. We'll have an overview of the NCERT followed by the solving of back exercises. This times where we can use, where there are good use of the microbes for us, and also this will give you a brief introduction about the about subject microbiology, which is a second year subject in EBS, a thorough discussion and about each microbe and bacteria, virus, fungi you'll learn in your second year itself, the diseases they cause, how to treat them, how to diagnose them, and etc. So here we'll discuss about in this chapter we will NCERT lines part by or ask the lines as it is. So let's start. In the first page, there is assets nothing. Basically, they have asked that which kingdoms have microorganisms. So that are Monera, protista, and fungi. Grammar fungi, bacteria and fungi can be grown on nutritive media to form colonies, which can be seen by naked eye. So here the word naked eye is possess a proper cell structure, so they can't be cultured on culture media. These are a few diagrams. This is a rod-shaped bacteria called bacilli. The spherical shaped bacteria is in coca. This is the bacteria, rod-shaped bacteria. This is 8.2b is a virus which causes respiratory illnesses. You need to remember this, and which is rod-shaped. So you need to remember the shape here. So these are just the petri dishes with. There are a few diagrams. This is the rod-shaped bacteria, which is called bacilli. So figure 8.1b shows the spherical shaped bacteria, which is called cocci. Then this is a diagram of the bacterium with a flagella. So in figure 8.2, we can see a few of the diagrams of viruses. A is bacteriophage. Bacteriophage are the viruses which infect bacteria. B is adenovirus, which causes respiratory illnesses. And C is tobacco mosaic virus, which is rod-shaped. You need to remember the shape of the bacteria. So here are the petri dishes. Here you can see there are growing colonies which can be seen with naked eyes. And you need to remember here that in petri dishes the culturing is done, or it is also called culture plate. Now let's discuss about the uses of bacteria in household products. First is the curd formation. So in curd formation, the bacteria used are the lactobacillus, and the group of the lactobacillus bacteria is called lactic acid bacteria, which is LAB. Now, how is curd formed from milk? It is on the basis of the milk has lactose in it. So lactic acid bacteria acts on the lactose, and they have an enzyme called lactase. So the lactase acts on the enzyme lactose, turning it into lactic acid, which leads to decrease in the pH, and therefore the milk protein casein coagulates, which leads to formation of curd. So as it is written in the theory, the milk protein such as casein, it coagulates. Now a small amount of curd is added to the fresh milk as inoculum. Just remember the word here, inoculum, which contains 1,000,000 of LOB. Now the advantages of formation of curd, or the advantages which lactobacillus gives is, first, it increases the nutritional quality by increasing the content of vitamin B12. Second, it is also good for our stomach as it plays a very beneficial role in checking the disease causing microbes. And third, it increases the shelf life. It increases the shelf life. Now further we'll discuss about some doughs. For example, you must have observed at your home, like when dosa or idli is made, when it is cooked, the dough is fermented. It is kept overnight and it is cooked in the next morning. So what happens, actually the yeast which is present, which comes from the atmosphere, here are a few questions given, like can you tell which is the metabolic process? It is fermentation. From where does the CO2 come? It comes from the substrate. And from where does these bacteria come? They come from the atmosphere. So the yeast present in the atmosphere, they come into the dough, and a few are and many are present already in the dough. So they ferment the dough overnight, and which leads to production of CO2 gas, which leads to the rising of the dough and the fermentation of the dough. So therefore, yeast is also called baker's yeast or brewer's yeast. Brewer's yeast we'll discuss in the next section. The name is Saccharomyces cerevisiae. So also there are many drinks which are formed through fermentation. For example, toddy, which is a traditional drink made from fermentation of sap from the palm trees. Then the toddy contains 4 to 6% alcohol. Then there is also fishes fermented, soya bean, bamboo shoots, etc. Now let's discuss about cheese. Cheese is one of the oldest food items. It is also, there are various types of cheese based on the type of organism used and the for the time for how much time it is processed, and which gives it different texture, flavor and taste. It comes from the microbes. You need to remember this. Microbes. So let's discuss about a few types of cheese. First is soft cheese, which has 45% to 85% water. The name is camembert cheese, and the microbe used is Penicillium camemberti. Then next is semi-hard, which has 45% water. It is called Roquefort cheese, and the microbe used is Penicillium roqueforti. Next is hard cheese. It has less than 40% water. It is called Swiss cheese and also called cheese with ice. Why it is called cheese with ice? Because it has large holes in it. That large holes are due to production of CO2. And then the microbe used is Propionibacter sharmini. Now let's see in the text. See, there is Swiss cheese. It has large holes. It is made through the bacterium Propionibacterium sharmini, and then there is Roquefort cheese. Now let's discuss the use of microbes in industrial products. We need to remember the word fermenter here, that whenever there is large scale production or the industrial scale production of any product or any item, it is done in fermenters when fermentation is required. Now let's discuss about fermented beverages. So you must be aware about various types of alcohols, which are various types of ethanols which are consumed. So on glucose, yeast acts through its enzyme zymes and leads to formation of ethanol plus CO2. So this is the basic reaction which happens, and you need to remember the enzyme as well as the reaction. So the various types of alcoholic beverages or the fermented beverages are wine, beer, whiskey, rum, etc. And these are also made by the yeast Saccharomyces cerevisiae, and therefore it is also called brewer's yeast. Now there are two types of drinks, like without distillation and with distillation. Let's discuss about these. So without distillation and with distillation, they basically differ in the quantity of ethanol present. So the distilled ones have higher concentration of ethanol, whereas the non-distilled one have a decreased amount, less amount of ethanol. So therefore they are called soft liquor and distilled drinks are called hard liquor. Now a few examples. In non-distilled we have wine, which is made from grapes, and beer, which is made from malted barley. You need to remember grapes and malted barley. And this is the average concentration of alcohol present in it. Now let's discuss about a few examples of distilled liquor. So there is brandy, which is distilled wine. Distillation of wine leads to formation of brandy. Whiskey, which is made from cereals. Vodka, which is made from potatoes. Rum. Gin, which is made from European rye scale cereals. And tequila. Now let's discuss about the antibiotics. The term antibiotic was coined by S. Waxman in 1942. You need to remember this. Now what is meant by antibiotics? Anti means against and bio means life. So therefore it is against life. Now antibiotics can be either bacteriostatic or bactericidal. Bacteriostatic means it stops the growth and bactericidal means it kills. Now the first antibiotic to be discovered was penicillin, and which was produced by Penicillium notatum, who founded Alexander Fleming. So what happened? Alexander Fleming was doing a few experiments in which he observed in one of his unwashed culture plates, the Staphylococci could not grow. So he noticed that nearby there was growing penicillium.
which was producing penicillin. Hence, there was discovery of antibiotic. But its full potential was established by Ernst Chain and Howard Florey. During the World War II, it was extensively used. So Fleming, Chain and Florey, they received Nobel Prize in 1945, and you need to remember this too. These are a few diagrams of fermenters and fermentation plant. Now, antibiotics have greatly improved the capacity to treat deadly diseases such as plague, whooping cough, which is called Kali Khansi, diphtheria, Galgotu, and leprosy, which is Kush Rog. You need to remember these names as these are asked commonly in the MCQs. Now, let's move on to the next section, which is chemical enzymes and their bioactive molecules. Now, here we'll discuss about Aspergillus niger, which is a fungi. Aspergillus niger, a fungus which produces citric acid. Then Acidobacter aceti, a bacterium, it produces acetic acid. Clostridium butylicum, bacteria, produces butyric acid, and Lactobacillus, a bacteria which produces lactic acid. Now here are 4 examples, remember it by heart. Aspergillus niger, Acidobacter aceti, Clostridium butylicum, and Lactobacillus, and the products they produce. Now, yeast, which is Saccharomyces cerevisiae, is also used in the commercial production of alcohol ethanol, as we discussed just earlier. So here is the 5th example. Yeast Saccharomyces cerevisiae, it produces ethanol. Now, microbes are also used for the production of enzymes. Till now, here was the examples of acids. This was examples of acid producing bacteria. This is acid producing bacteria. Then we discussed for ethanol, which is alcohol, and now we'll discuss about enzymes. For example, lipases, which are used for detergent formulation, are helpful in removal of oily stains. Then we'll discuss about juices. In juices also, juices are very clear. The bottle juice which we buy, these are very clear, and these are made clear by using of these enzymes which are called pectinases and proteases. They dissolve the pectin and the proteases contained from the plants in the fruits, and they make the juices clear. So the bottled juice are made clear through these enzymes. So next we have streptokinase. Streptokinase is produced by the bacteria Streptococcus and is modified by genetic engineering and is used as a clot buster for removing clots from the blood vessels. This is very important. You need to remember this. Streptokinase clot buster, and it is used for myocardial infarction to cure myocardial infarction. Actually, myocardial infarction can't be cured because it is an irreversible change. It is death of cell, but it can be stopped from for happening further by removing the clot. So the 3 to 4 enzymes which we discussed are lipases, pectinases and proteases, and streptokinase. So these are the enzymes. And then here we have alcohol. You just need to mark your NCERT search so you can read it just in a half an hour to 1 hour. That is the tactic to read the NCERT. Now next we will discuss about cyclosporin A, which is also very important as it is an immunosuppressive agent. And it is produced by the fungi Trichoderma polysporum, which is a fungus. You need to remember this. Now next we have statins. Statins is a lipid lowering agent. It is used when there is high cholesterol, when patients have high cholesterol or lipid function, there is defect in the lipid function. So these are blood cholesterol lowering agents. How does this act? It acts by competitive inhibition, it acts by competitively inhibiting the enzyme responsible for synthesis of cholesterol. Hence, they lower the body cholesterol levels. And when there is lipid dysfunction or there is dysregulation of the lipid levels in the body, statins are given, and statins are usually given during the night, as during the night time it is the time when most of the cholesterol synthesis takes place. Now next we will discuss about the sewage treatment. Now in the sewage treatment, now you need to remember this line. The municipal wastewater is called sewage. The municipal, the municipal wastewater is called sewage. Sometimes you are asked the example of sewage. So you can write this as it is. Or a proper definition should also include the organic matter and the BOD. It's called sewage. Now, where is this treated? It is treated in the sewage treatment plant, STPs. Now, treatment is done by, there are various steps in the treatment of the sewage. So we'll discuss about this. There are many heterotrophic microbes present in the sewage. So you need to remember this. And this treatment is carried out in 2 steps. First is the primary, in which there is physical removal of the large particles through filtration and sedimentation. For see here, filtration, sedimentation and physical removal. And subsequent filtration is again done after the removal of floating debris. And after that, this is called primary sludge. When then the grit are removed by sedimentation, all the solids settle from the primary sludge and the supernatant forms the effluent. So all the solids settle down during the sedimentation is called the primary sludge. The settled solid is called primary sludge, whereas the supernatant is called the effluent. The effluent from the primary tank is taken to the secondary tank, so which is treated further for which is biological treatment. So in the primary effluent. Now here also the effluent is the primary effluent. You need to remember this because after that there is also called something secondary effluent, which we'll discuss. Now there is primary effluent. It is passed to large aeration tanks, which mechanically agitated mechanically and air is pumped into it. Why does air is pumped into it? Because it has a lot of aerobic microbes which are turned into flocs. Now you need to remember the term flocs here, which is very, very important. Flocs, these are masses of bacteria associated with fungal filaments to form mesh-like structures. It is kind of something like there are bacteria, and there is fungi in it, fungal filaments. It is embedded in fungal filaments. So this is called flocs. Now after the sedimentation, see first there is filtration. Filtration happens after the removal of floating debris. For example, there are floating debris. From here, after filtration, removal of floating debris, it is lead to a sedimentation tank. This is the sludge, and this above is the primary effluent. This is sedimentation. And this is sludge. So it is then taken for secondary treatment, which is biological treatment. So in the biological treatment, air is pumped into it and there are flocs present in it. In the flocs, the microbes consume the major of the organic matter. They use the organic matter as the substrate and they grow, and this helps in reduction of the organic matter. So now here comes the term biological oxygen demand. Biochemical oxygen demand. See, as I made a mistake here, you guys are also very, very prone to make a mistake here. You need to remember this is biochemical oxygen demand. Because in many MCQs, there will be asked full form of BOD. It is biochemical oxygen demand. See, bio you can remember through the microbes are used and chemicals since reactions are taking place, that is consumption of the organic matter. So just remember it by breaking it. Now, after the microbes consume a significant part, there is significant reduction in BOD. Now, of the effluent. Now what is BOD? Here is the definition of BOD. BOD refers to the amount of oxygen that would be consumed if all the organic matter in 1 liter of water were oxidized by the bacteria. Now the words you need to focus here are 1 liter of water and oxidized. So BOD refers to the amount of oxygen that would be consumed if all the organic matter is in the 1 liter of water were oxidized by bacteria. So these are the few words you need to focus in on this line. So this line can be asked as it is in the MCQ or in your school exams, and you need to be very correct with these words. Now after sewage water is treated till the BOD is reduced, then the BOD is measured. The BOD test, how it is done by the BOD test. What is BOD test? It measures the rate of uptake of oxygen by microorganisms in a sample of water. Thus, BOD, rate of uptake of oxygen by microorganisms in a sample of water. Thus, this is also a very important line. Thus, BOD is a measure of organic matter present in the water, indirect measure of the organic matter present in the water. Greater the BOD of wastewater, more polluting it is, more polluting potential it has. This is also important. Greater the BOD waste, more polluting potential it has. Now, once the BOD is reduced, then the sediment, now then it is again allowed to sediment, and this sediment here is called activated sludge. Why it is called activated sludge? Because it has bacterial slogs in it. Now, bacterial slogs in it. Now, some part of the activated sludge is pumped back to the aeration tank to serve as inoculum. As we studied in the formation of curd, I mean, always an inoculum is needed to start the process. So, a small part of the activated sludge aeration tank is pumped back into the aeration tank to serve as inoculum. Now the remaining part of the sludge is pumped into large tanks which are called anaerobic sludge digesters. Anaerobic sludge digesters, and these are used in the formation of biogas, which is a source of energy as it is inflammable. Now what happens is that these bacteria anaerobically digest the remaining sludge, anaerobically digest bacteria and fungi, and this can be used as a biogas since they produce a mixture of gases which is methane, hydrogen sulfide and carbon dioxide, and can be used as a fuel. Here you need to remember the gases methane, hydrogen sulfide and carbon dioxide. And the word activated sludge is very important. Now the effluent formed after it is the secondary effluent which is released into the rivers. See, the effluent from the secondary treatment is generally released into the natural bodies like rivers. Now, after some time, I would also like to mention that there is a tertiary treatment or the chemical treatment where the water is treated using chlorine treatment is given to the water sometimes when it has to be used or purified more or cleared more. Now, let's discuss about an environmental fact here. That is, Ministry of Environment Forest has initiated a Ganga Action Plan and Yamuna Action Plan to decrease the pollution in these rivers as it is increased to a greater extent due to the industries and other anthropogenic actions. Now let's move forward. The microbes in the production of biogas. So biogas is a mixture of gases as just discussed, mixture of gases, but predominantly it contains methane and how it is produced by microbial activity as a fuel. Now here what you need to focus is the type of gas produced depends on the type of organism and the substrates, microbes and the substrates. So there are two factors in microbes and the substrates. Now the fermentation of dough and the cheese making, the main gas was produce CO2 because but however there are certain bacteria that can grow anaerobically on cellulose material and produce large amount of methane along with CO2 and H2O, and these are called methanogens. Methanogens, they produce large amount of methane along with CO2 and H2O. So for example, one such common bacteria is methanobacterium. These bacteria are also commonly found in anaerobic sludge during the sewage treatment, or these are also present in the rumen of the kettle. This is a part of the kettle stomach. The kettle stomach has four parts, out of which one part is rumen, where the digestion of cellulose takes place. Breakdown of cellulose takes place. Now the excreta, the kettle dung, which is common, is also rich in these bacteria. That's why it is also used as a for the generation of biogas, which is called gobar gas. Now a few facts about the biogas plant. It is a concrete tank which is 10 to 15 feet deep, and biowaste materials are collected. Slurry of dung is fed. Then there is a floating cover. I'll tell you this through the diagram more, which is more important. Now also here a more point you need to note here. This marsh gas. It also has CH4, that is methane. Now we'll just discuss through the diagram. See, this is from here the sludge is feeded. The sludge will be feeded from here, and the sludge will be feeded from here. After the feeding of the sludge, it will go into the digester. Now this is a floating chamber, gas holder. As and when we will feed the slurry of the dung, it will go into the digester. The methanogens or the other bacteria will anaerobically respire, and these will lead to production of gases, and the accumulation of gas will take place at the upper part of the digester. Since the gases are lighter in density, they'll move to the upper part of the digester, and which will lead to upward movement of the floating chamber. And this can also indicate the amount of biogas produced in the digester, and will lead to starting and stop of the biogas plant. From here, the gas is taken out in a pipeline and is supplied to the households or to the plant where it is needed. From here, the anaerobic sludge is also accumulated in this separate tank, and it can be used as a fertilizer. So this is a typical layout of the biogas plant. Now, the digestion here takes place in 3 steps. The point which we need to focus on, it takes place in 3 steps. First step is digestion. 3 steps. So the first step is hydrolysis, which is breakdown of substances using water. Then there is acidogenesis. Acidogenesis, it is done by facultative anaerobic bacteria. Facultative anaerobic bacteria. Now the third step is methanogenesis. It is done by methanogens, as we read just now. Some of the examples are methanococcus, methanobacterium, and methanobacillus. These are anaerobic bacteria. Now let's form a chain reaction for it, which will be complex polymer. Then first step will happen, which is hydrolysis. It will lead to formation of simple monomer. Then the second step, which is acidogenesis, it will lead to formation of acetic acid. And the third step, which is methane CH4. So this is just the layout of it. Now next, it can be used as a fertilizer. Then biogas plants is mostly built in rural areas and can be used for cooking and lighting. Now here, one word, Due to the efforts of Indian Agricultural Research Institute and the Khadi and Village Industry Commission, it has been promoted. Now you need to remember these two names. Now also the digestion of the cellulose takes place at a very slow pace. Therefore, it is the rate limiting step of the biogas plant, and you need to remember this. And this is asked in the MCQs previously. Cellulose digest digests at very slow rate. Mm, Therefore, rate limiting step. Biogas production. Now next is microbes as biocontrol agents. So biocontrol agents, like there have been many problems to control the weeds and the and the eating of crops. Here we'll just see biological methods for controlling plant diseases and pests. So increasingly chemicals have been used for it, but these are very harmful to humans as well as for the plants and if we consume them, and it can also lead to soil pollution and water pollution. So what are the biological controls for this? It has been that uh, the more variety a landscape has, the more sustainable it is. Now in the organic farming, a system is created. The insects, which are called pests, are not eradicated, but instead they are kept at a manageable level. Basically, they protect the food web which is present and the ecosystem which is present. Instead of erasing all the insects, it can lead to a disturbance in the food web and which can lead to a disturbance in the ecosystem. So they don't eradicate it, they keep it at a manageable level in the ecosystem, contrary to the other conventional practices. Now after that, the eradication of the creature are often described as pest and as the eradication is often, but it has a beneficial predatory as and the parasitic insects which depend on them as their food and hosts will not be able to survive. See here, uh, the beneficiary predatory and the parasitic insects which depend on them as foods, they will not be able to survive. So if you eliminate all the insect causing disease to the plants, after that the parasitic insects or the predatory animals or the birds which feed on them, they'll not be able to survive, and this will lead to decrease in their population also. So this will develop this will, if we make every organism in a manageable level, it will lead to a appropriate means of biocontrol. Now let's discuss a few examples here. For example, the ladybird, dragonfly are used to get rid of aphids and mosquitoes. Then we have examples of butterfly. Another bio that can be introduced to control butterfly caterpillars is Bacillus thuringiensis. They control the butterfly caterpillars. This is a very common example. It will be explained in detail later. So these are available in sachets as dry spores, these are available in dried spores and which are mixed with water and sprayed onto the vulnerable plants, such as plants of brassica and fruit trees. And when they're eaten by the insect larva, the gut of the larva, these are in the Bacillus thuringiensis spores are intaken by the larva, and in the gut of the larva they release toxin, and the larva gets killed. The bacterial disease would kill the caterpillars but leave other insects unharmed. So this is a targeted approach. Other insects will be unharmed but kill the caterpillar. So this, because of development of these methods, genetic engineering in the last decade so has introduce Bacillus thuringiensis gene into the plants. So this is called BT cotton by the help of genetic engineering. Now next, a biological control for the treatment of plant diseases, fungus, Trichoderma. Trichoderma is a free-living species and it is present in the root ecosystem and has a very effective control in the controlling plant pathogens. Now next we have viruses as pathogens are pathogens that attack insect and other arthropods. So majority of these are used as biological controls and in the genus nucleopolyhedroviruses. These are used excellently for species-specific narrow-spectrum insecticidal applications. They have no negative impact on mammals, birds, and fishes. Now, these are used under the program Integrated Pest Management Program, where eco-sensitive areas are treated. So overall, here you need to remember are a few examples. For example, uh, ladybird and dragonflies. Then next is Bacillus thuringiensis, and the third is baccalovirus. The third is Trichoderma, and the fourth is baccalovirus. These four examples. Now next, as the usage of biofertilizers, in organic farming, most of this bacterium can be used as organic farming as the source of various nutrients. For example, the nitrogen, which is very commonly studied, it is the rhizobium for the roots of leguminous plants. The rhizobium has a symbiotic association with the roots of leguminous plants. The main sources of biofertilizers are bacteria, fungi, and cyanobacterium. So the first example is rhizobium. These bacteria can fix the atmospheric nitrogen into the organic form, and a few examples of these are Azospirillum and Azobacter. Now next we have fungus mycorrhiza, which have a symbiotic association with the plants mycorrhiza, and many members of the genus Globus form mycorrhiza. Symbiotic associations. Now the fungal symbiotes in these associations absorb phosphorus from the soil. Now see, here it was atmospheric nitrogen, now this is phosphorus from the soil. And these associations have also benefited the plant as such as resistance from root-borne pathogens and increased tolerance to salinity, drought, and overall increase in plant growth and development. Now, just you need to remember these 2-3 points. Now the last is cyanobacterium, which are autotrophic microbes and are well distributed in the aquatic and terrestrial environments. A few examples are Anabina and Nishtha. What they do, they fix the atmospheric nitrogen and are used as an important biofertilizer. The blue-green algae is also added to organic matter to increase the fertility of the soil. So here you need to remember these three. So here we are done with the chapter. This was an overview of the chapter. This is a summary. As such, there is no extra point in the summary, but do give a read to it once. We'll just quickly skim through each chapter and discuss the practice exercises again. So in the first phase, there is just the naked eyes point which you need to remember. That the colonies can be seen with the naked eyes. These are a few diagrams. Then next we have the formation of curd. From here you need to remember the B12, vitamin B12 and the procedure how the curd is made. You need to understand it. Then the Saccharomyces cerevisiae, yeast is used and the name that has. Then there are various types of cheese. You need to remember all these various types of cheese and the microbes used in it. And the characteristic texture, flavor and taste depends on the microbe used. Now for large scale production, fermenters are used. Then these are the types of drinks made, non-distilled and distilled, hard liquor, soft liquor, their examples and concentrations, and the products from which they are made, the raw products. Now the reaction enzyme diamase is used for the formation of fermented beverages. Now we move on to antibiotics. It was coined by S. Waxman. First antibiotic was penicillin, founded by Alexander Fleming. Then Ernst Chain and Howard Florey gave a full use of it. It can be bacteriostatic or cidal. A few examples of the diseases you need to remember here. Then the chemicals and enzymatic uses. The acid-producing bacteria are Aspergillus niger, Acetobacter aceti, Clostridium botulinum, and Lactobacillus. Then for alcohol, yeast is used. For enzymes, there is lipase, pectinase and protease, and streptokinase, which is used as a cross buster for and can be used in ML. And then the bioactive molecules, which is cyclosporin A, it is an immunosuppressive produced by Trichoderma polysporum, which is a fungi. And then there is statins, which is produced by the yeast Monascus purpurus, and it is used as a blood cholesterol lowering agent. Now here, how the questions are asked in this from this paragraph, they'll give you match the followings. In one column, there will be the name of organism, and in the next column, there will be the name of uh, the substances they produce, and you need to match them accordingly. So you need to remember them properly. They also sometimes change the species name behind. So you need to take care of that since we need to stick to the NCERT. Then we have the sewage treatment. It is done in STP, sewage treatment plants. There, it consists of two processes, primary and secondary. The primary house, in the primary treatment, first is primary treatment. In primary treatment, first there is filtration. From the filtration, floating debris are removed. Then they are passed for sedimentation. In sedimentation, the settled down is called the sludge, and the supernatant is called the primary effluent, which is then sent for biological treatment. In biological treatment, we'll just form a flowchart here. Biological treatment. First, the primary effluent. It is passed through aeration containers. Where flocs are present. These flocs, they after these flocs are the masses of bacteria with fungal filaments to form mesh-like structures. Here, the biological oxygen demand is decreased. Air is pumped into the container, and BOD of the primary effluent is decreased. After that, it is sent to a secondary to anaerobic digesters. Anaerobic sludge digesters. Now let's make a diagram only. See the primary effluent which comes. Here flocs are present. These are flocs. So after the digestion, this is aeration tank. From here, the activated sludge is taken out, which is settled at the bottom. This is anaerobic digester. This is anaerobic digester. So we'll show that it is sealed from the top. And this is used for biogas production. Now from here, the primary effluent which is treated, it is released into the water bodies. So the treated treated one is released into the river. Sludge. Now this is called activated sludge. You need to remember this word activated. Now these are the names of the two plants which are used. Then we have production of biogas. It is through the methanogens, which are the methane gas producing bacteria. Now also the methane biogas, it contains these three gases which you need to remember: methane, hydrogen sulfide and carbon dioxide, and which is predominantly methane. It can be used as a fuel. Now next we have the steps of biogas production. First is hydrolysis. Complex polymers are changed to simple monomers through hydrolysis, then to acetic acid through acidogenesis, and then to methane through methanogenesis. Now, breakdown of cellulose is a very slow step. Therefore, it is called the rate limiting step of biogas production. Now, next, the biogas production was increased in India through Indian Agricultural Research Institute and Khadi and Village Industry Commission. Now, the microbes is biocontrol agents. Here we have ladybird and dragonfly, which reduce the aphids and the mosquitoes. Then we have Bacillus thuringiensis, which decrease the butterfly caterpillars, and it is introduced into the cotton plant and leads to formation of BT cotton through genetic engineering. Then we have fungus Trichoderma. Trichoderma is present in root ecosystem. Then we have Baccalovirus. It is from the genus no, it it is a control agent for the genus Nucleopolyhedroviruses and also attacks insects and other arthropods. Like the Baccalovirus genus is Nucleopolyhedrovirus and they attack insects and other arthropods. These are species spectrum, narrow spectrum and insecticidal. These are used in ecologically sensitive area. Now, biofertilizer, there are three examples: rhizobium for nitrogen fixation, mycorrhiza for phosphorus fixation, and cyanobacterium for nitrogen fixation. Now, we are done with the chapter. So, let's discuss the questions. So, in the first question is that you can see in your surroundings the presence of bacteria. You can take any sample for the first question. It can be soil or water and you'll see the presence of various microbes in it. And next are the various gases which are released during metabolism. This can be either methane or CO2. You can read for this is biogas and this is for fermentation. So this is done, this is done. Next, where will you find the lactic acid bacteria and their useful application? Obviously, curd formation. Now, next we'll go to traditional applications from some food items. From wheat, there is formation of bread and cake. Rice, there are formation of South Indian food that is dosa, idli, uttapam. Then we have Bengal gram, dhokla and khanwan made from wheat. Then next we will have in mushroom disease and their examples you can see. Then next we have two species of fungus and the antibiotics produced. First can be penicillin, which is produced from Penicillin notatum, and next is cephalosporin, which is produced by Cephalosporium erimonium. I'll just write it down. Cephalosporin. Cephalosporium erimonium. And next example is penicillin, which you can check in the text by Penicillium notatum. So what is sewage? As discussed in the chapter, sewage is the municipal wastewater is called sewage. It is also called sewage, but a proper definition will include by BOD also. So in what ways sewage can be harmful to us? Obviously, it can cause water pollution and other soil pollution, and it can lead to very many diseases due to the pathogenic microorganisms present in the sewage and also other waste products present in the sewage. Now, what is the key difference between the primary and the secondary waste? We'll just discuss about the key points here. First, primary and then secondary. The primary one it is on the basis of physical treatment, but it includes biological treatment. It is based on sedimentation and filtration. It is based on the biological digestion, biochemical, I would say. Biochemical digestion. Now next, it is less expensive, less and less time consuming, whereas it is more expensive and more time consuming. And then it is less complicated, whereas this is more complicated. Do you think the microbes can be used as a source of energy? Yes. How? Biogas. Just read through the chapter how biogas production is done and then make the diagram here. Then microbes can be used to decrease the use of chemical fertilizer and pesticides. Yes, they can be. How? For example, the biofertilizers can be used. For nitrogen, rhizobium and cyanobacteriums can be used, and for phosphorus, the fungus mycorrhiza can be used from the gene chlomus. And now there are three water samples: river water, untreated sewage water, and secondary effluent. Now the effluent which was released in the river is called a secondary effluent as explained during the chapter. We can see here the flow chart. So this is the primary effluent after the primary treatment, and after the secondary treatment which is formed is the secondary effluent. And it is released in the rivers. Now the undigested sludge is the untreated one, and the river water is present here. So these are the three we are talking about here. First is the untreated sludge here, then second is the secondary effluent, and third is the river. So accordingly we'll arrange. Now the BOD demands are given. It is 28 and 400. Now, what they have asked, assuming that the river water is relatively clean. So, since they have given that the river water is relatively clean, so we have read that if BOD is high, then pollution potency or the pollution cause is high. So, since river water is cleanest, so river water will get a BOD of 8 mg per liter. So, river, which is A, it will have 8 mg per liter. Now B, untreated sewage. Untreated sewage is the most dirty, most polluted of it. So it will be 400 mg per liter. And then C, it is secondary effluent. It will be 20 milligram per liter. Now find out the name of the from the microbe from which cyclosporin A, an immunosuppressant, is formed. As given in the chapter, it is Trichoderma. Trichoderma. And statins, it is obtained from Monascus purpurus. Now the role in the following: single cell proteins. Okay, single cell proteins are used for the protein requirement of the human body. So the various examples of single cell proteins. This is also covered in your previous chapter, which is the strategies for enhancement in food production. So the single cell proteins are examples of blue-green algae. I'll just make down a list here. Blue-green algae, which is spirulina. Then algae, which is chlorella. We have bacteria, which is methylophilus and methylotropus. Then next we have fungus, which is fusarium. These are few examples of SCPs. SCPs are used for increased intake of protein for humans. Since in paneer there is, for example, when you consume paneer, which is also rich in protein, or uh, chicken or non-veg, which is also rich in protein. But for vegetarian diet, which are the substances which are rich in protein is fungus, is a single cell protein. Now for soil, you can just discuss in soil the role of microbes. For example, then there are fertilizers. Then the pathogens, you can discuss the pathogens present, and so on. Now we have to arrange the following in their importance order. So according to that, what will be the order? See, biogas is used as a fuel. Citric acid is used as a preservative. Penicillin is an antibiotic, and curd is a food. So first should be penicillin, since it is an antibiotic, which it saves human lives. Then next is biogas. It is used as a fuel. Fuel is the Without fuel, nothing can be done. Obviously, everything is powered through it. And then third is the curd, which is our food product which we eat. And fourth is citric acid, which is used for preservation. It is just an order. You can also form an order on your basis, but there should be an logic to it. Then how do biofertilizers enrich soil? For example, the nitrogens and the phosphorus, rhizopus and blue-green algae. And then for phosphorus, we have glomus. For phosphorus, we have glomus, as we discussed here, or mycorrhiza. The rhizobium, they fix the atmospheric nitrogen. Mycorrhiza, they fix phosphorus, and cyanobacterium, they also fix nitrogen from the atmosphere. So with this, we are done with the chapter and the back exercise excels. I hope you had a good learning experience, and do give a read to the chapter once or twice so that you remember the lines very well and have no problem in solving the MCQs. Do remember them by heart so that all the lines of the NCERT are learned by heart, and there is no error or incorrect question which you make in the exam. All the best.